I'm audible? Yeah. Good afternoon. First, I want to thank Sheba for inviting me to this meeting. When I got the invitation, I got the impression it was also in honor of the Nobel Prize this year on the circadian clocks. And I want to, it's not about only the animals that you meet in research, but also about people. And I had the chance to meet two interesting persons working in circadian clocks before I myself started working in circadian clocks. And the one guy is this, Jeff Hall, and maybe Sheba talked about him. And Jeff Hall is very outspoken. And particularly if you want to study kind of genes involved in behavior, he already pointed out in 1993 or 5, what is my soul neurine protein doing in the ass end of the fly, as well as in the brain, where I first found it and where I wanted to be. And it means that basically most of the time you will not find a gene for behavior. You will not find a gene for religion. You will not find one gene for intelligence. All kind of genes are making proteins that are involved in cellular kind of processes. So kind of genes that you are finding in the brain, or kind of like, I was in a lab, we were searching for genes that are kind of have something to do with behavior. And most of the time we got kind of genes that were involved in cellular processes. And everyone was kind of disappointed. Everyone wanted to have a kind of a specific learning gene or something like that. So this is about the pleiotropy of genes and proteins in cellular processes that can occur in different tissues. And one of the tissues that you have is the brain. And so this is a guy, you should kind of YouTube him and look how he is talking. It's very impressive. And then there's Vijay Kumar. And I have to say that I only met him kind of like 5 to 10 times. And so I don't know him very well. But he kind of like introduced me to some of the insect science community in India. And the only thing that what I can say about him was when I kind of first was in his office, and I hope I remember it right, and Sheba might kind of like confirm it, I saw a picture of this guy. This guy is Konrad Lorenz. It's a German zoologist. And he got a Nobel Prize together with Karl von Frisch and Nico Tinbergen in the 1972, I think. Interesting thing was that zoologists of my age in Germany wouldn't probably hang a picture of Konrad Lorenz for several reasons. And I'm pointing this out that I had to kind of somehow come to India, not to hear about Konrad Lorenz, but to be reminded about this person. This person is Aschoff. And he's one of the, Professor Aschoff, he's one of the founder of the circadian clock. And actually when, so we came via kind of some kind of strange way to study in circadian clock in honeybees. And the first chat I had about our data with Vijay Kumar, he said to me, look at a paper, Frisch, this is not Karl von Frisch, Brigitte Frisch and Aschoff, in 1985. They did similar experiment that you did. And I have to say, I had the paper on my computer, but I was not aware of that anymore. And I will tell you later about this paper. So I kind of like, I just want to kind of honor him or memorize him. And thank you him for kind of like when we started to go into circadian clock research. So one part of the research that we are doing or establishing to do in my lab is kind of studying the molecular neurobiology of daily foraging behavior. Why kind of foraging behavior in honeybees? And that is, I have to tell you, that's the most important behavioral paradigm to have identified the capabilities, the sensory and cognitive capabilities of insects. And this is due to a kind of a phenomenon of the natural behavior. And we have kind of like in honeybees foragers, and these foragers kind of over the whole day are flying back to a specific food source and food location, collecting food and bringing it back to the hive. Under natural condition, kind of a single forager does probably about 15 foraging trips to that specific location. This behavior, which we call flower constancy, and which has also a spatial constancy, actually kind of provides the researcher with the opportunity to train honeybees to a feeder. And training honeybees to a feeder is a paradigm that Carl von Frisch introduced into kind of research. And using this behavioral paradigm, he kind of identified color vision in 1914, dance language in 1920, this is almost 100 years ago. Then kind of in 1929, honeybees were used to show for the first time that animals have a sense of time. And then kind of like later, in the 50s, sky polarization, landmark counting in 1995 by a different group, and even in 2001, kind of that they have a kind of proto-cognitive capabilities of concept formation. The point is that if you can train an animal to a feeder and the animal comes back to that feeder in a regular time, then you can ask the animal a question at that feeder. For example, you can present kind of the sugar water, which is the reward to come back to the feeder, above a blue cardboard. And you can present water kind of like with non-rewarding above a yellow cardboard. And then you can kind of train the bees, and after some time you can ask the question, are you preferring the yellow cardboard or the blue cardboard? And this is what Carl von Frisch in 1919 did, and he kind of experimentally, behaviorally demonstrated that honeybees have color vision. Physiologists at that time kind of expected insects not to have color vision. So daily foraging behavior. So if someone is saying honeybees are intelligent, then they might be, but more importantly is these are the insects which we can demonstrate that insects have maybe intelligence or have kind of like complex learning forms. Because we can train them to a feeder and we can ask them a question. And so this is, and then till the 1950s, with this system, honeybees were kind of like what we today call model systems to study behavior. And now what my lab tries to do is to use molecular tools to understand what might happen in the brain while the bees are foraging. And we do it in that we kind of do the behavioral experiments, and then we catch the bees, flash freeze them, with that kind of like all kind of molecular processes stop. And post-mortem, we can use molecular tools like mass spectrometry or gene expression to identify what might have happened or what might have happened molecularly in the brain when the bees did this behavior. The assumption is that if we kind of compare, if we kind of identify changes in the molecular titers or gene expression in the brain occurring during a specific behavior, then these molecules or genes might be involved in generating that behavior. And the goal for us is the identification of molecular processes in neuron populations involved in higher or more complex behavioral responses or capabilities. Kind of capabilities and responses that you hardly have a chance to test in Drosophila. Certainly, this approach is correlative, and we cannot kind of do causal experiments. So now the question is kind of like, I have the foraging behavior, how do I get kind of to identify kind of like interesting molecules? And we had kind of like two approaches. One is the question, does daily foraging affect clock gene expression? And here we are kind of like in the research of circadian clock. And the second approach, and I will explain it later, does foraging behavior involve an induction of gene expression? We tested that. So the first part of my talk, does daily foraging affect clock gene expression? So 1932 already, Wahl kind of trained honeybees to a feeder, foraging at a feeder. And on the left side, he presented the feeder the whole day. And on the right side, he presented the feeder only in the afternoon. He did this training for 10 days in a row, presenting kind of feeder either the whole day or either only in the afternoon. And on the 11th day, he tested what the bees are doing. There was a feeder, but the food was not presented. So these are just flying out, and he counted which bees are coming close to the feeder. And what you see here is then the daily activity rhythm, kind of foraging rhythm. You see kind of like maybe a peak in the early morning, some midday peak he found, and some evening peak he found. And if he's kind of training them only to come to the feeder from 3 to 5 p.m., you see that their peak is kind of shifted at 5. You don't see hardly any activity in the morning and in noon. You see a little bit of activity starting in noon, which kind of circadian rhythm people would call anticipatory behavior. Yeah? So you can kind of shift foraging activity by kind of presenting the feeder at a specific time of the day. There's a training involved, kind of like over 10 days. So basically, we did now the same kind of like, what was it, 70 years or no, even more years ago further. We did the same experiment, and because the genome is sequenced, we can kind of like ask kind of like how gene expression changes occurred during this training. So we did the same. We did ad libitum feeding. We had a training in the morning, a training in the afternoon, 10 days training. And again, on the day of a collection, the feeder was presented, but there was no food. And and then we kind of tested whether this has an effect on kind of genes of the molecular clock. And that's kind of like a very good bet. So I don't want to go into the circadian clock. There are many kind of neurons. In honeybees, we just only know kind of like 4 or 5 so far. Most important is here that kind of like 2 major clock genes are cycling, showing a strong cycling. Kind of this cryptochrome-2, it's kind of has peaks in the night, early night, and pair also kind of like peaks strongly in the night. And we are using these 2 clock genes to kind of see whether the feeder training or the restricted foraging has an effect of the cycling of these 2 genes. Remember, it's low during the day, high in the evening. And we had the 3 kind of different training procedures. And here you see, so it's important. So here you see the daily cycling of the 2 genes. I just go through cry. So as you see the cycling at libitum feeding, the kind of like feeder was presented the whole day. And this cycling, kind of like with a peak in the night, is very similar to what has been described before in the literature. Then if we train them, if you have trained the bees to come to a feeder in the morning between 8 and 10, what we see is a slight shift to the left. I indicated here at the minimum. It's kind of like, so you see that's kind of like a little shifted to an earlier time. And if we train them in the afternoon time, we see a shift more in to the evening time. And here's a summary. Hope you can see that. So morning training or restricted foraging only in the morning, or foragers that have been restricted to foraging only in the morning, compared to foragers that have been restricted to foraging in the afternoon, they have molecular clocks that are in phase different, and they show a shift. Yeah? It means kind of like similar to kind of like you're kind of like going to the west or so, and then kind of you have your jet lag. Then you need to reshift your molecular clock to be on that time. So foragers trained in the morning are on a different time, so to speak, compared to foragers that are only foraging in the afternoon. And a point I have missed to make is that we did these experiments under natural light dark conditions. Yeah? So it kind of, and we think that the natural light dark cycle is the strongest zeitgeber of the circadian clock and the molecular clock. And here, kind of like in the presence of the light dark cycle, we even kind of are able, by kind of restricted food presentation, to shift the molecular clock. Our data are confirmed by kind of the study that Vijay Kumar suggested me to read, Fritsch and Ashraf, 1987. What they did, they kept a honeybee colony in a flight room where you had constant light and where you had constant temperature. And they were trying to see whether restricted presentation of food kind of entrains the activity of the colony. And you see here, first you see kind of like activity over the whole day. And then here starts restricted feeding for 2 hours. And you see a shift of the kind of flight activity of the foragers, or kind of like a concentration of the foraging activity during the time of the food presentation. And he already kind of said, under the influence of periodic feeding, the activity of a honeybee colonies has the characteristics of entrined circadian system. So he already kind of described the phenomenon. We now show that actually food presentation under natural light-dark cycle, which he kind of avoided to have, also leads to an entrainment of the molecular clock. Second, an experiment in Gene Robinson's lab was done published in 2011, where they trained foragers of the same colony, one group in the morning to feeder A, location A, and kind of the other group to kind of feeder B in the afternoon to location B. And then they were comparing these phenotypes. Don't want to get into detail. They only measured 2 time points. And what they found, so these are the morning trained, and then they kind of measured cry2 in the morning and in the afternoon. And what they found is that cry2 in the morning trained is a little bit higher than in the morning. And in the afternoon trained, cry2 is higher in the morning and kind of lower in the afternoon. These are kind of actually corroborating our result. And we did kind of like a more complicated study where we kind of measured the titers at different time points. And so they showed only this difference by only using 2 time points. If you kind of like take 6 time points, you kind of really show that the cycling of the genes is shifted. This is to make the point that, if you are going for gene expression is kind of like a phenomenon in time. So probably if you want to kind of identify genes that kind of are involved in the behavior, you should test kind of gene expression over several time points, and not just kind of like take a snapshot. And kind of the approach of Gene's lab is like having phenotypes and taking snapshots. Yeah. So, but molecular processes are not happening in a snapshot. So to summarize this part, is that we have demonstrated that restricted foraging under natural light-dark conditions is capable to shift the brain molecular circadian clock. Food to reward, I haven't shown you kind of those results. Food to reward alone is sufficient to induce this shift. Together, the behavioral experiments by Frisch and Ashraf, as well as our molecular studies, kind of indicate that foraging activity, I have to say foraging activity of food collection. The interesting thing is that honeybees that are foraging, they don't feed the food. They're collecting the food and bringing it back. This is kind of for the circadian clock people, and it's a little bit different than food entrainment. So foraging activity or food collection can entrain the circadian clock in honeybees. And interestingly, there was a study by a lab in Germany that kind of demonstrated, they were kind of observing a colony, and they were seeing that there are actually bees that are preferentially active in the morning and bees that are actually preferentially active in the evening. And that this has a kind of genetic basis. So this is something what's in human research known as chronotypes. So it appears that, that what we did was learning. We were presenting the food at a specific time. They were learning and then adjusting their foraging. But it appears to be that even in division of labor between foragers, there are kind of chronotypes, one that are active in the morning and one that are more active in the evening. And this has a genetic component and probably the genes that we have tested. So then the second approach is, does daily foraging behavior involve kind of induction of a genomic response or induction of gene expression? That means kind of like when the bees are starting to forage, kind of do we see kind of a specific induction of gene expression? And for that, the idea is going back to what's called the genomic action potentials. Kind of in the brain, you kind of have the action potentials, but actually kind of high kind of neural activity or prolonged neural activity leads to kind of gene expression. And this gene expression is kind of mediated by the secondary molecular pathway and the induction of so-called early immediate genes. These are transcription factors that are the first genes that are expressed. They are expressed within kind of like, here you see it, half an hour of activity. And these unusable transcription factors are used already kind of for several years to identify neuron populations that are active in the behavior. For example, in bird song, kind of like if a bird learns a song, kind of people kind of gave the tutor, it's kind of listening to the song, and then they are sacrificing the bird and looking in the brain, looking, kind of identifying which area was active during the song presentation. And second, of course, because it's transcription factors, if you find a transcription factor that's activated during foraging, you kind of use this transcription factor to identify downstream genes that are induced by these transcription factors, and then test, for example, whether it's kind of involved in learning or something else. We used a transcription factor immediate early gene that's called early growth response gene. This is kind of like a test if you kind of cold seizure the animal, then you see an expression over the brain kind of increasing within 45 minutes and then decreasing. Here's the honeybee brain. You have the optic lobe, the central brain, and then here the mushroom bodies. And here is some kind of gene expression of this EGR1 that's done by my student. Why did we take EGR1? It's identified in honeybees, it's identified in Drosophila. It has a kind of story about learning and memory in vertebrates. And interestingly, it has been demonstrated by Gene Robinson's lab that it is one of kind of 4 or 5 transcription factors that are actually regulating the transformation from nurses to foragers. So it already has a developmental function kind of to induce genes that or regulate genes that might be involved in foraging. So here now we did a similar experiment again, kind of 10 days of training. We trained these bees to come to a feeder between 2 and 4 o'clock, and then for 10 days again. And then we did on the day of collection, the feeder was presented, different to the circadian clock, because we wanted the bees to kind of keep foraging on that feeder. And what we did is then we did kind of a collection before foraging, and then we did a collection during foraging, and then a collection after foraging. And during foraging, we did the collection of every 50 minutes. And that then kind of like shows here the results. This is the projection. This is only for the foraging time, starting at T0. That is when we are kind of presenting the food for the first time. And then we kind of collected bees at 50 minutes intervals. And we were looking in the whole brain, kind of the expression of this immediate early gene, EGR1. And what we see is that starting with continuous foraging, we see kind of like an increase of this gene, like an increase in the gene expression in the brain. And particularly also what's important is that it is kind of like a maintained gene expression. If you have only a single stimulus, you have kind of like it rises to 30 or 45 minutes and then it goes down. Yeah? And this is a different gene, and I don't want to go on this. So starting, so a bee that's coming to a kind of feeder, and it's starting kind of to forage, collect, fly back to the hive, comes back to the feeder. There we see, doing this behavior, we see it kind of like a molecular response in the brain. Then, and that is for the first time, people did similar experiments already with honeybees, but what we did was now ask the question, so if this transcription factor is upregulated, does this upregulation also kind of lead to the upregulation of candidate downstream genes? And we were lucky that at the time when we did these experiments, kind of people came up with ideas about downstream genes. And we tested a kind of a series of downstream genes. And this is dopamine receptor 2, ecdysteroid receptor, dopamine ecdysteroid receptors, dopamine decarboxylase. And so this is handpicked because we already know that there's evidence that these genes are involved in learning and memory in insects. And then we kind of took these 2 genes, which are involved in kind of general cellular processes and kind of like cellular homeostasis. And for a few genes, and these are 3 colony replicates. And for example, here, this is the dopamine receptor. We see an increase of the dopamine receptor during this foraging activity. Yeah, so There's not only kind of like an increase in the expression of this immediate early gene, but also kind of downstream genes are activated during this foraging. And you have to remember that I told you that this same behavioral paradigm was used to show that honeybees can learn colors, yeah? That honeybees learn time. So this is a kind of behavioral assay for which it has been demonstrated that it is inducing learning, or that, how to say, that during this behavior learning is occurring. Now then we ask, because kind of we did always whole brain kind of gene expression, we ask, can we identify kind of like neurons in the brain that are kind of expressing these molecules? And already from previous study, it was known that actually EGR1 is kind of highly expressed in this region, the mushroom bodies, which is kind of like the region for learning and memory and multimodal integration. And just to kind of show you that, so the Scully says that all the neuron population of the mushroom bodies, and kind of people already kind of describe that this population can be separated in different smaller groups of population. And particularly, I just want to kind of mention these violet ones. These are the so-called small canyon cells. They get mixed input. EGR1 is expressed, USP, HR38, and dopamine. We tested all these genes, and these genes are upregulated during foraging. And we did, again, the same training, 10 days, then collection. We did a collection T-minus 30 before foraging, T0 when the food was presented, T90 after 90 minutes, and after foraging. And then my student Aritni compared using in situs gene expression in the brain areas. And the first that she found was that here's a T0, and here you see the Kalitzes, and you see that here that mainly she sees at T90 an increase in the gene expression of EGR1 in the area of the small canyon cells. Still, there's also in the larger cells some response, but strong response in the small canyon cells. So we can say there are maybe also other brain areas, but how to say, the qPCR measurement of expression changes that we are measuring are also happening in the mushroom body, brain areas in the honeybee brain that are involved in learning and memory. So my second summary is successful foraging is accompanied by a genomic response involving transcription factors like EGR1, HR38. We tested some others. They don't show an increase. Then the EGR1 response is accompanied with an upregulation of candidate downstream genes involved in learning and memory, as well as cellular homeostasis. And it is the first study that suggests that foraging-related upregulation of EGR1 mainly occurs in the small canyon cells of the mushroom bodies. I don't know what the time I'm going on. So then there are two additional questions, and in historical order. So one is, does feeder training affect EGR1 expression? So what I have showed you is that the EGR1 goes up when the honeybees are starting to forage. Now we are asked do we see actually, after 10 days of training, even an increase in this gene when the bees are not foraging? And again, same training procedure. And so we had ad libitum training, morning, afternoon training, and then we were collecting. And the idea was now to see in the bees that have been trained for specific time points on the 11th day when we are not presenting a feeder, where do they show EGR1 expression? They are just flying at the moment. And what we see is during ad libitum feeding, and they are not foraging during this time, we see an upregulation over a longer range of upregulation almost over the whole day with a peak at 2 o'clock. If we have trained the bees from 8 to 10 over 10 days, then we see an upregulation of that gene at the time of the training. And if you have trained them in the afternoon, we see an upregulation of that gene at the time, or again on the time of the training. So we think that this is evidence that just time training might have an effect of the expression of EGR1. But how to say, the bees were still flying during this, so maybe it's the activity. So what we then kind of did was kind of we had a time training, and then we were kind of using artificial rain. And artificial rain makes the bees not going out. And so they are staying in the hive. So then we were asking, after 10 days of training, what are the bees doing when they are not flying out? Are they still expressing? Are they still upregulating this gene? And then kind of like here, so we did collections, kind of like before kind of training, and these are the training time from 2 to 6 o'clock. We did several time points here, before and during the time of training, the same with the morning training. So here you see, I'm kind of like talking about this, for example. So we see kind of like, so the bees have been trained, but they're not flying. We are collecting them from the hive. And what we are seeing is that actually there's an increase of EGR expression, a smaller kind of increase, but a significant increase in the gene expression of that gene, a little bit before and at the beginning of the time of training. We see this in kind of 2 experiments, and we also see this in the morning. And this kind of like reminds us of the anticipatory behavior. If you remember my first slide, kind of like Van Waal showed, when you're kind of training the bees to come at a feeder from 4 to 5 or something 4 to 6, you see already activity kind of before. And this is what kind of animals learn. If you kind of like how to learn kind of a specific behavior at time, if you connect this time, you are actually kind of getting active a little bit earlier. You yourself kind of can see it when you are kind of training yourself to wake up at 8 o'clock. You first kind of use the alarm kind of to put it at 8. And after several days, you will wake up kind of like 20 minutes before, even before kind of the clock was ringing. Yeah? And we think that, so that is a hypothesis, that this kind of increase of EGR1, which is little compared to the increase that we get with active foraging, is a kind of like maybe a molecular, kind of an anticipatory response in molecular processes. Okay, then the question was again, kind of how to say, we measured kind of like this in whole brain gene expression, kind of where is the gene expression doing? And my student did here kind of like in situs, fluorescence in situs. DAPI is a cell staining EGR1, and here DAPI EGR1 compared. And I hope it's kind of like seeable that before foraging you see hardly any expression. This is foraging, doing active foraging. And you see expression here in the small canyon cells. Here you see it. And here this is a rain experiment. Kind of again, they kind of were not flying. They were in the colony. And we see kind of a gene expression in the same kind of group of cells. Yeah? So to summarize, it appears like EGR1 shows a small upregulation in time-trained foragers when they are prevented from leaving the hive. This EGR1 upregulation appears to start before the time of feeder presentation. EGR upregulation occurs in the same neuron population as during active foraging. And as I said before, we hypothesized that EGR1 upregulation might be some kind of anticipatory molecular response similar to anticipatory behavior. And the last experiment is that we tested. Now, we have always used one kind of feeder. And so we were asking what is happening with the EGR1 expression if we train kind of bees to 2 feeders, to visit a feeder in the morning and in the afternoon. And we train them from 8 to 10, and then from 4 to 6. And here I think we have 57. What was the 57? So one is the same. So what you have actually is you get 3 groups. You get bees that are only visiting the feeder in the morning. You get bees that are only visiting the feeder in the afternoon. And you get bees that are visiting both feeders. And here's the results. So those bees that are only visiting in the morning, we see an upregulation of EGR1 in the morning, kind of, or we see a, let's say, higher expression in the morning compared to the afternoon time. In the bees that visit the feeder in the afternoon, we see a higher EGR1 expression in the afternoon compared to the morning. And for the bees that are coming to kind of both feeders, we see an upregulation at the time of the both feeder time training. We don't see it clearly going down, which kind of might be kind of like some other problems. But more importantly is that they show an increase of EGR expression as both time feeders. So that means that kind of EGR1 might be linked to the time. So that is my last slide. Maybe I don't know whether I'm over time. So what I have demonstrated here is two phenomena. One is that I hope that the circadian clock people are agreeing with me. It's about entrainment. Entrainment means kind of like that the behavioral activity, physiological activity is kind of like adjusted to a specific light-dark or kind of feeding cycle. And we kind of, that is known that the light-dark cycle kind of leads to an activity cycle, which makes kind of the bees flight at a specific time. And also we know that there are many genes in the brain as well as in all kind of tissues that are regulated by this circadian rhythm, this light-dark cycle. What we demonstrate now that in honeybees, that the food or foraging activity has a strong effect and can affect flight activity as well as clock-dependent kind of molecules, right? So the point is kind of like how to say, for example, also for kind of like saving energy. So let's assume that foraging is learning. Then you kind of, for example, try to express the genes that are kind of involved in learning to the time when you are actually foraging, the learning processes are happening. But that is kind of shifting the whole activity rhythm. And we have different groups, probably we have in honeybee colonies, different groups, foragers active in the morning, which are on different time, and foragers in the afternoon on different time. And then we have a phenomenon, and this is about time memory. So at what time a specific thing occurs that has to be done. And it appears that, I mean, I'm not saying that we have strong evidence, but maybe we have a way to identify it. It is when kind of EGR1. We know that kind of learning a flower, visiting a flower, kind of like has an effect on EGR1 expression. And we see that if this is this correlated at a specific time, we see even that kind of without having the flower, without kind of like flying out, kind of EGR1 already shows a regulation that's probably kind of regulated by the circadian clock. And this could be kind of like the possibility of that might be kind of like a molecular process that has something to do with time memory. And then EGR1 kind of induces genes that are involved in these learning processes. Here you have all the physiological processes kind of like that are regulated by the circadian clock, and here you can have learning of time. That is the end of my talk. Thank you very much.